All right, so in this video, I'm going to be going over the 2015 FRQ. This is number 2. And this one is going to be, I would have to say, somewhat similar to what you might see on this year's exam, just because it takes some specific content, but a lot of analytics going on with this FRQ. So with this one, it gives you pictures of each of the main steps in cell respiration, and then you have a couple of questions as well as a little bit of evolution tied in. So let's kind of just take a look at this first animation. It kind of gives you a really simple overview where glucose gets broken down into pyruvate. Pyruvate is then oxidized even further into acetyl-CoA. Then it gives you the Krebs cycle over here, where you take that acetyl-CoA, combines with oxaloacetate, and this is the whole Krebs cycle or citric acid cycle. Make some GTP, but the key thing is you make a lot of NADs and FADs later to be used on the figure 3, which is on the electron transport chain. So that's kind of significant to understand. Then it kind of walks you through the process of respiration, and you have to explain each step. So it talks about cell respiration includes the metabolic pathway of glycolysis, the Krebs cycle, and the electron transport chain as represented in those figures. In cellular respiration, carbohydrates and other metabolites are oxidized, and the resulting energy transfer reactions support the synthesis of ATP. Using the information above, describe one contribution to each of the following in ATP synthesis. We got to explain how the catabolism of glucose in glycolysis and pyruvate oxidation. You have to explain how oxidation of those intermediates in the Krebs cycle contribute to ATP synthesis, as well as the formation of a proton gradient in the electron transport chain. So let's go back to this picture real quick. And just really quickly off to the side, I'm just going to draw a simple mitochondria so you guys understand and remember kind of where all this is taking place. So remember that if this is the cytosol, this is where glycolysis takes place. And then it goes into the mitochondrial matrix, and this is essentially where the Krebs cycle takes place. I'll just put a K there. And then right around here, really on the inner membrane, all throughout the mitochondria, this is where the ETC takes place. So just to kind of show you, that's where the Krebs cycle takes place. In glycolysis takes place outside in the cytosol. The electron transport chain is taking place right there. Okay. So now we kind of have that, and basically what it's wanting us to do is to do an explanation of how each one of these is going to be contributing into ATP synthesis. So let's kind of take a quick overview for glycolysis, and it kind of does a couple of key things here that helps explain this out. So there's really 2 things you could state for part A in terms of catabolism of glucose. So catabolism is referring to the breakdown of glucose. How does that result in the formation of ATP? Well, a couple of things here is that glucose makes ATP right here. So that's one contributing factor, and that refers to substrate level phosphorylation. And if you don't remember what substrate level phosphorylation is, I have a picture right here to remind you of what that actually looks like. So substrate level phosphorylation looks like this, where, excuse me, basically some ATP is produced that's not efficient. So it basically means that you're taking a phosphate from something else, you're using an enzyme, and you're actually putting that phosphate onto ADP to make ATP. So big thing is ATP is produced, but it's not efficient, and a phosphate is removed from a substrate. So in fact, when you get into glycolysis, that splitting, you're taking in some ATP to make a huge output eventually with all the NADs and FADs. So here's kind of a really 10 or 11 step reaction where you actually take glucose, your end product is going to be pyruvate, and this is showing you there's a couple of steps here involved where when you get in here, step 10, ADP makes some ATP. That's an example of substrate level phosphorylation. It takes a phosphate from this molecule, this phosphoenol pyruvate, it takes that phosphate off and puts it onto ADP. And that's your substrate level phosphorylation, okay? And the biggest thing too, when you get into the rubric here, is when you go back to this animation, you could state that you do make some ATP this way, via substrate level phosphorylation. It's produced out here in the cytosol. But a big thing that hopefully you all are familiarized with is NAD is produced, NADH. And NADH is going to later be used to power the electron transport chain. So if you're thinking of how you should formulate your response for this one, let's go back here on really what this should look like. So when you're writing your response, I would say using the information, you could have stated one thing. It makes ATP via substrate level phosphorylation. Or you could have stated that NADH is produced. And that NADH is going to be used for the electron transport chain. So either one of these would have got you 1 point. So if you stated that ATP via substrate level phosphorylation or NADPH is, or not, excuse me, NADPH, just NADH is produced for the ETC. So that's how you got 1 point. Let's look at the Krebs cycle or the citric acid cycle. How does this contribute to the formation of ATP? Well, we can talk about a couple of things here. Let's just kind of remind ourselves where this is at. Notice now we're actually inside the mitochondria. It doesn't include the link reaction here, but you could state that you do make some ATP via substrate level phosphorylation. I've seen the POGL say that it's ATP. It's technically GTP, but it's going to be working honestly the same way. But you could also state you produce a high output, a very, very high production of NADH and FADH2, and these will all be used for the electron transport chain. So I would state that what is the point really of the Krebs cycle? You make a little bit of ATP or GTP in this case via substrate level phosphorylation, but the key thing is you make a high production of so high produce of NAD and FAD for the FADH2 for your electron transport chain again. So that's one huge contributing factor. And then this last one is probably the most critical to understand. I don't think you necessarily had to go crazy in depth in terms of your explanations here, but let me just erase a few things here. So if you're thinking about the electron transport chain, kind of the whole payoff is those protons. So let me kind of jump back to my notes and just kind of revisit this. When you look at your electron transport chain, the focus really should be those electrons. So let me just kind of remind you guys of this. So notice we're on one of those inner membranes, and when you take a look at this closely, always kind of think backwards. Well, where did that NADH come from? And remember that there's another membrane here. So it takes place in between the two membranes of the mitochondria. So NADH is actually going to dump off its electrons, and those electrons are eventually going to be shuffled. The electrons end up on oxygen and make water. So the key thing is that you want to understand that these electrons are pumping protons against their gradient, right? You build up this electrochemical gradient. So basically the whole point is you want to build up all of these protons in between the two membranes just for those protons to diffuse back down through that ATP synthase. And as this ATP synthase protein is spinning, it literally takes a phosphate and forces it onto ADP, uses some mechanical energy to make this high output of ATP. So basically, simply put, as long as you have these protons diffusing down through ATP synthase, and that protein is spinning because it's like a turbine. As long as these protons diffuse down, causes this protein to spin like a turbine. As long as that's spinning, it's taking phosphates and ADPs and putting them together. A lot of people get confused, and just what I like to tell them here, you have to just assume that the mitochondria has an abundance of ADP and phosphates just there. So you have a high production of ADP and phosphates just there. As long as this thing is spinning, it's forcing them together. Now, as these protons diffuse back down, some of those hydrogens go onto oxygen to make some water. But the key thing here is that this process is known as chemiosmosis. So it's really osmosis of chemicals. That's the easy way that I remember this as osmosis of what really the chemical in this case is those protons. So as those protons diffuse down, ATP synthase spins, phosphate is attached onto ADP, and you make ATP. So I always kind of advise you guys to think backwards. Well, where did these protons get here? Well, NADH provided the electrons to force them against the gradient. Well, where did NADH come from? Thinking backwards, NADH was produced in the Krebs cycle as well as glycolysis. So let's get back to the FRQ. In this FRQ, what we needed to explain in this case was really formation of the proton gradient. How does that contribute to ATP synthesis, or excuse me, ATP synthesis? Is that the protons diffuse through ATP synthase, and that produces, so protons diffuse through ATP synthase, that produces ATP via chemiosmosis. So that process of chemiosmosis are those protons diffusing back down. So basically this is just showing you as these protons get built up. It doesn't look like it shows what's happening here with ATP synthase, but technically there would be ATP synthase here and these protons would diffuse down. So that was really kind of the specific that they were wanting you guys to get out, that if this is ATP synthase, as these protons diffuse down, that provides that energy to put a phosphate onto ADP. So A is worth 3 points. So this one was pretty specific, no doubt, but they kind of gave you the visual on that one. For B, it wants you to use each of the following observations to justify the claim that glycolysis first occurred in a common ancestor of all living organisms. So basically you just got to back up these statements on why, how did this actually, how can you provide some specific content to justify this claim. So they made the claim that all existing organisms perform glycolysis. Okay, so basically you want to understand, well, why has that happened? Why do we all cells, if it's eukaryotic or even prokaryotic, why do they still do glycolysis? And so there's a couple of things that they gave you for the rubric here. So if all of them performed glycolysis, the justification is a little bit of evolution and natural selection in the sense that we all have the gene. That means that there is some form of gene to make the enzymes. And those enzymes that we all share allow it to do glycolysis. So it served as some benefit that gene, that those genes that we have, right? So the DNA that we would have been passed down to make the enzymes. Those enzymes are allowed to be for glycolysis to take place. So it's a really weird thing to think about when you consider everything that we are capable of. But when you think about the genes that we share with really all species, if you go back to this slide on glycolysis, really, there's enzymes involved, and here's the list of all the enzyme names for each one of these. When you take biochemistry in college, you're most definitely going to have to memorize each one of these. But when you think about what's happening here, is this step one requires an enzyme, right? So there was some gene that was expressed to make that enzyme, right? And let's say that this has another enzyme shape here. Again, there was another segment of DNA that was read to make that enzyme. And then so on and so forth. Maybe this one has an active site shape, looks something like that. So basically, this first enzyme involved is hexokinase. Hexokinase, there's a gene that we all share, whether that's prokaryotic or eukaryotic, even in plants. This hexokinase enzyme is the same for the most part among many species. So there's a reason why that served as an advantage and that's been passed down. So when you go back to this rubric, hopefully this makes a little more sense, that gene to make all those enzymes for glycolysis has been passed down. And when you get to the second one, the second claim, how do you justify that glycolysis occurs under anaerobic conditions? And this is something that was pretty specific to really the Miller-Urey experiment, but the key thing is that there was no O2 in early atmospheric conditions. So that Miller-Urey experiment, if you forgot, remember that they basically replicated early atmospheric conditions and it tested positive for proteins and nucleic acids after a while. But the big thing is that you can do glycolysis in all cells and all conditions. So the key thing is all cells, all conditions can do glycolysis. So, I mean, excluding, I should say temperature and pH, but whether there's oxygen or not, glycolysis is gonna happen. You can make some ATP via substrate level phosphorylation, all right? Now, this last one is kind of interesting to discuss, but how can you justify this claim that a glycolysis occurs only in the cytosol? So this one was really dealing with the endosymbiotic theory, and that is something that I think is really, really specific for you guys to have gotten this point. The glycolysis predates cell types with membrane-bound organelles. So if you forgot what the endosymbiosis theory is, it is this, that this was your ancestral cell. This was essentially the mitochondria. It essentially engulfed a prokaryotic cell that was really, really good at doing some form of respiration, and that's how it got that second membrane. So in terms of this cell could only do glycolysis. So all of the main key big payoff phases in the mitochondria how it got that double membrane was as it got engulfed, it received that double membrane from that endosymbiosis. So other evidence includes that the DNA in the mitochondria, it's circular, which kind of gives evidence that is an ancestral prokaryotic cell. But the DNA in your mitochondria is different than the DNA in your nucleus. So key thing is that when you go back to this, glycolysis occurs outside the mitochondria. So that means that in early ancestral times, we could only make ATP via glycolysis because we didn't have a mitochondria. So that's really what your explanation should have been for that one. So that's how you got those 3 points for part B, by justifying it in some way, shape, or form, one of these three. Now, when you get to C and D, C for sure, I would bet is something that you would probably have to do on your exam. And let me explain why. Because this one, it takes a concept that we really don't discuss a lot, and there really would be no way of knowing how this works. It's really just taking your analytical skills and seeing how you can apply it. So a researcher estimates that the complete metabolism of glucose produces 30 molecules of ATP. For each molecule of glucose. The energy released from the total oxidation of glucose under standard conditions is 686 kilocalories per mole. The energy released from the hydrolysis of ATP to ADP, an inorganic phosphate, is 7.73 kilocalories per mole. So it's wanting you to calculate the amount of energy available from the hydrolysis of 30 moles of ATP, calculate the efficiency of total ATP production of 1 mole of glucose in the organism, describe what happens to the excess energy. So this one has a lot going on, but when you kind of take a step back and really analyze it, it's not too terrible. So let's try our best and make this make sense. So I'm going to make a little template here. So for C, we'll solve it down here. So basically this means that 1 molecule of glucose can actually release 686 kilocalories per mole, meaning that if you have glucose in a lab setting, it can actually give off that amount of energy. So 1 glucose molecule can actually release 686 kilocalories per mole, okay? 1 ATP can actually provide, according to this rubric, 7.3 kilocalories per mole. So 1 ATP can actually release 7.3 kilocalories per mole. So it wants us, other key things is that when you actually break down glucose in cell respiration, you make 30 molecules of ATP. So in respiration, okay, 1 glucose can actually make 30 ATPs. So 1 glucose in respiration can actually make 30 ATP. So the first thing it's wanting us to do is to calculate, we got to calculate the amount of energy available from the hydrolysis of 30 moles of ATP. So if I have, and it's going to count, the units are going to cancel out, so moles will cancel out. But basically, if I have 30 molecules of ATP, So if I have 30 moles of ATP, and I multiply that by the energy that is going to be released, it tells us that we're going to have 7 point. So we're going to multiply that by 7. Let me just redo this. So if I have 30 moles of ATP, and I'm going to multiply that by how much energy is released for 1 mole of ATP, it's going to be 7.3 kilocalories per mole. Moles cancel out. And basically all you do is you multiply 30 by 7.3 and you're going to get your value here. So I'm just going to do 30 times 7.3. And I got a value of 219 kilocalories. So that basically means if I can produce 30 molecules or 30 moles, I should say, of ATP, it's going to release that 219 kilocalories. So let's take a step back and look at this first, not this first, but what I wrote up here. It tells you in the problem that in cell respiration, 1 glucose can actually make 30 ATPs. So, but 1 glucose makes 30 ATPs. 30 ATPs actually can provide 219 kilocalories. Now, the next thing that I want you to know is calculate the efficiency of the total ATP production from 1 mole of glucose. Well, it tells you earlier that 1 mole of glucose can actually release 686. So that means glucose, in theory, should make 686 kilocalories per mole. But you're only in respiration producing this much. So for the efficiency, so we're going to calculate the efficiency, and it's a simple percentage problem. I'm going to take that 219 divided by the amount that glucose does give off and multiply that by 100, 686 times 100%. So in this case, I will get a value of, now I'm not including units because they'll all cancel out, 219 divided by 686 times 100%. It's going to give me a value of 31.9%. So let's just kind of take a step back and analyze what we have written so far. So to get those 2 points, you really needed to make sure that you understand the specifics on what you just calculated out. So for this one, it looks like we have this 219 is going to give you 1 point. This 31.9% is going to give you your second point. The last thing that we have to explain is describe what happens to the excess energy that is released from the metabolism. So basically, we should be getting 686. We actually only get 219 from 1 molecule of glucose. It's only about 31.9% efficient. And the key thing on this one is where does the rest of that energy go? And this one is really getting into ecology, but the key thing is that anything breaks up, you're going to lose a lot of it as heat. So you're going to lose a lot of that energy as heat. So it's not going to be super efficient because you're always losing some amount of energy to heat. So those were going to be those 3 points for that one. All right, so that's going to be your 3 points. This one was another 3-pointer. So 3 points for this one. So you're already up to 9 for that. Then it says the enzymes of the Krebs cycle function in the cytosol of bacteria, but among eukaryotes of enzymes, they mostly function in the mitochondria. Pose a scientific question that connects the subcellular location of the enzymes in the Krebs cycle to the evolution of those eukaryotes. And that kind of just leads into what I already talked about, that these bacteria cells, remember that if you just had a ancestral bacteria cell, that these were where the enzymes were in their cytosol, right? So they had enzymes that helped kind of break down cell respiration. And that's really where all those enzymes are. But basically it means now that when we look at our cells, if this is I'll put quotations around this, that this is your prokaryote cell, okay? It's saying that the enzymes are just in the cytosol. But when we look at our cells, it's saying that these same enzymes are found in our mitochondria. So what would be a good scientific question? Really pose a scientific question that connects the location of the enzymes of the Krebs cycle to the evolution of the eukaryotes. I would state something along the lines that is there evidence that the genes are the same that help support this endosymbiotic theory? That if the enzymes are the same, is there similar DNA? And that helps support the endosymbiotic theory. So Just to kind of wrap this one up, let's double check the rubric on this one. So there is the yeah, the heat is released here. Yeah, so does it suggest that mitochondria were once prokaryotes? Yeah, I think that's kind of talking along the same lines that if those were the same, were they eventually at one point in time their own prokaryotic cells? So that is the wrap up on that particular FRQ. If there's any other concerns, I think this one for the most part was pretty vague, but yeah,